Lecture 3: Eukaryotic chromosomes and chromosome biology in eukaryotes. The genome is partitioned in two sections called chromosomes. Each chromosome is composed of a complex and poorly understood arrangement of DNA and proteins, chromatin. Each chromosome also contains a set of functional elements necessary to ensure that DNA is efficiently replicated and partitioned into daughter cells at cell division. Replication is initiated at DNA replication origins. At the ends of chromosomes, specialized structures, telomeres, maintain the integrity of chromosomes. At cell division, the centromere and kinetochore facilitate chromosome segregation. In general, repetitive and so-called junk DNA is packaged into highly compacted chromatin, heterochromatin, whereas gens are found in less tightly packaged euchromatin. The human karyotype, the number and appearance of its chromosomes with human chromosomes. Cytogeneticists have utilized a variety of staining techniques to generate maps based on an invariant banding pattern. G-bands are revealed by staining with the diacyled GMSA after treating mitated chromosomes with a protease to digest some of the proteins using this method. Whilst darker regions of the chromosome contain DNA with the low +C content and generally replicate late in the cell cycle. In contrast, R-bands, reverse GMSA banding, identified by GMSA staining after heat treatment of chromosomes, contain G+C rich DNA and replicate early. The normal pattern of G and R banding gives a reference map, and even now, cytological analysis of human chromosomes can be very useful for tracking down disease gens. For example, many cancers are associated with specific chromosomal deletions, and some diseases have been tracked down due to particular translocations, swapping bits between chromosomes. The functional elements of eukaryotic chromosomes replication origins. Every chromosome needs a place, or places, at which DNA replication can be initiated. Clearly these must be tightly controlled, used on lines per cell cycle. The structures of some bacterial replication origins are well understood. The best characteristic eukaryotic replication origins are the yeast autonomously replicating sequences, RS, which bind a complex set of ORC proteins, origin recognition complex, that act to control the initiation of DNA replication. See Dr. Moni's lectures found that staining. 19. In contrast to yeast, very few metazone replication origins have been defined. However, it is clear that the situation is more complex than in yeast, and, while many of the proteins that associate with origins are conserved throughout eukaryotes, the sequence motifs identified in Cerevisiae Rs are not found in origins in other eukaryotes. In addition, in multicellular eukaryotes there appear to be many more potential origins than are actually used by any one cell in any given S phase, and which origin is used can vary during development. For example, in Drosophila early embryos, when cell division is very rapid, each chromosome replicates from multiple origins. As the cell cycle lengthens later in development, the number of origins used per chromosome appears to decline. Thus, there is developmental modulation of origin activity. It is also clear that different parts of the chromosome are replicated at different times, in early, mid or late S phase. Research to define the nature of replication origins and their control is ongoing, with current evidence indicating that factors such as DNA topology, nucleosome density, chromatin and transcription state all contribute. Telomeres in eukaryotes. The DNA in each chromosome exists as a linear molecule, in contrast to most bacteria where the chromosome is circular. This raises an interesting problem at the ends of the chromosomes. DNA replication requires a primer, short stretch of DNA with the free 3O group, but at the very end of the chromosome this doesn't work. Therefore, at the end of every cycle of DNA replication, progressively more of the terminal DNA will be lost. Again, look back at Dr. Moni's lectures. So why don't chromosomes vanish? Chromosome ends are protected by special terminal structures known as telomeres, which contain arrays of short G-rich repeats. For example, in Cerevisiae there are 300 bits per second of 5 Tg, 1-6 Tg, 2-3, -3 feet, n, while in human there are around 2-20 kb of 5 TTAGGG 3 feet, n. These G-rich repeats help stabilize the ends of chromosomes by providing multiple binding sites for several DNA sequence-specific binding proteins, and forming a DNA secondary structure called the T-loop, that tucks the DNA end safely away. When telomeres eventually become too short, cell death or senescence is triggered. A telomere lengthening enzyme, telomerase, extends the chromosome end by adding more copies of the species-specific G-rich repeat onto the 3O group of the single-stranded overhang. The extended overhang then acts as a template for conventional lagging strand synthesis. In this way, a buffer zone of simple G-rich repeats is maintained at the chromosome end. Telomerase is a ribonuclear protein, RNP, complex that has both reverse transcriptase activity, that is, it is an RNA-dependent DNA polymerase, with its own inbuilt RNA template. Telomerase acts to top up chromosome ends in immortal cells, and its activity is present in unicellular eukaryotes and in the germline. and stem cells, of multicellular eukaryotes. Most cancers are associated with inappropriate telomerase activity 20 centromeres after replicating the DNA. Chromosomes must be separated during mitosis, such that each daughter cell receives the full complement of chromosomes. To achieve this, chromosomes are coordinated with the mitotic spindle via their centromeres. These are specialized structures on each chromosome where a large number of proteins assemble, including the centromere-specific histone H3 variant CENPA, to give a multi-protein complex called the kinetochore. The kinetochore is the structure through which the chromosome interacts with the microtubules of the mitotic spindle. This kinetochore-mediated link between the chromosomes and the microtubules allows sister chromatids to be pulled to opposite poles of the mitotic cell during anaphase. The first centromeres to be characterized were those of the budding yeast Saccharomyces cerevisiae. Budding yeast centromeric DNA is a 125 BP sequence with an at-rich core. However, this type of centromere, very small with a strict DNA sequence dependence, has turned out to be very unusual in eukaryotes, and the S. cerevisiae centromere does not function outside budding yeast. To digress, by combining the DNA elements required for origin, the Rs, telomere and centromere function in S. cerevisiae, it is possible to generate an artificial chromosome that will be stably maintained alongside the natural chromosomes in budding yeast. These Y-yeast artificial chromosomes, YACs, proved to be very useful cloning vectors, allowing the creation of genomic DNA libraries in yeast cells. Such YAC libraries were used extensively in the Human Genome Project, since they allowed large DNA inserts to be cloned, 500 kb, including DNA rich in repeat sequences, often poorly represented in bacteria-based BAC libraries. However, it should be noted that YACs do not function as chromosomes outside budding yeast cells. In contrast to budding yeast, the centromeres of the fission yeast Saccharomyces pombe are much larger and more complex. In this case they range in size between 35 and 110 kilobits, up to 3% of the chromosome, and are composed of a complex arrangement of a unique central core flanked by innermost repeat DNA, that shows a mirror image symmetry about the central core, which is in turn bordered by many, variously arranged, copies of two types of outer repeat DNAs. The large and repetitive DNA nature of S. pombe centromeres is very different from that found in budding yeast. 21 most poorly characterized are vertebrate centromeres which, since they often consist of mbps of repetitive DNA sequences, have not yet been fully sequenced. For example, human centromeres are composed of long tandem arrays of 171 BP alpha satellite repeats. We have even managed to generate human artificial chromosomes, HACs, by introducing large stretches of cloned alpha satellite DNA into cultured human cells. Genome composition repetitive DNA, various classes transposable elements. As we saw in Lecture 1, the human genome contains a considerable amount of repetitive DNA. It turns out that this can be categorized into various classes, dominated by types of transposable element and their deleted derivatives, copies of which are scattered throughout the human genome. Chief amongst these, and occupying a staggering 20% of the human genome, are the long interspersed nuclear elements, lines. These are retrotransposons 6 kilobits long, although most copies in the genome are truncated versions, 1 kb. They contain two open reading frames, ORFs, encoding an RNA-binding protein and a protein with reverse transcriptase and endonuclease activities. Cellular RNA polymerase II can transcribe full-length 6 kilobits line elements embedded in our genome, and the transcript generated serves two functions. Firstly, it is translated, alongside other mRNAs. The proteins produced remain associated with the line RNA itself, as a ribonucleic protein complex. This RNP complex enters the nucleus, where the line RNA then serves as a template for its own reverse transcriptase. This generates a DNA copy of the line RNA, which ends up inserted randomly back into the genome. Most of the lines in the human genome are partially deleted and no longer transpose, are no longer active, but a small number of elements of the line 1 subfamily, 100, are active. It is estimated that a new germline line 1 insertion arises at a frequency of 1 in 200 live births. 22 Hitchhiking on the enzymatic activity of the lines is the second largest family of genome repeats, the signs, unsurprisingly, short interspersed nuclear elements. These small repeats are transcribed by RNA polymerase II and use line-encoded enzymes to produce a DNA copy of the sign RNA, which is then inserted back into the genome. The most abundant signs in the human genome are the ALU repeats. They contain a recognition site for the ALU restriction enzyme. The line reverse transcriptase machinery can also occasionally mistakenly use a mRNA originating from a protein coding gene as its template, and this DNA ends up being inserted into the genome as a processed pseudogene, a DNA copy of the parental gene's mRNA sequence. A third family of retrotransposons is the long terminal repeat, LTR, like sequences, which resemble retroviruses. Like lines, LTR-like sequences can encode reverse transcriptase, POL, but very few of those in the human genome are active, that is, most are incapable of mobilization. A final class of transposon repeats is represented by elements that move via a DNA-based cut-and-paste mechanism utilizing the DNA transposon-encoded transposase protein. These contribute only 3% of the human genome, and there is no evidence of any DNA transposons being active in the human genome over the past 40 million years. They appear to be inactive relics. Altogether, repeats originating from reverse transcriptase copied RNA, lines, signs, LTRs and processed pseudogenes, or from, now inactive, DNA transposons, occupy 40% of our genome. Simple sequence repeats, SSRs, also known as satellite DNAs, are perfect, or imperfect, tandemly repeated non-coding DNAs that make up 6% of our genome. They can be subdivided into microsatellites, minisatellites and satellites based on the size of the individual repeat unit, monomer, and the overall length of the tandem array. See table. A variety of sequences are repeated, and they are generally written in the form NN, N, where N equals the nucleotides and N equals the number of times the motif is repeated, that is, GT, 6 equals GT 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 GT, ACT, 4 equals TACT TACT TACT. The alpha satellite DNA found at human centromeres, monomer size 171 BP, array size, often several Mbps, is an example of a satellite DNA with a clear function. There are several other satellites in the human genome, beta satellite and satellites II and III, which also occur in very large MB arrays and are heterochromatic and for which, as yet, no function has been identified. The 0.120 kilobits arrays of TTAGGG, N that form our telomeres can be classed as minisatellites. One of the most common microsatellites is the CA repeat, where N is between 10 and 100. Most simple sequence repeats, microsatellites, minisatellites and satellites, are polymorphic in the population, meaning that there are multiple alleles at each repeat locus. For example, a CA, N repeat at a particular location in the genome may be present at N equals 15, 16, 17, 18 and so on. In practice, this means that a given individual will be likely to have different alleles on each chromosome, thus tracking particular regions of the genome by the transmission of SSRs is possible, and b. Individuals are likely to have different spectrums of SSRs if a number of loci are compared. As we shall see, polymorphic markers are incredibly useful for mapping diseases and other traits. They are also useful for DNA fingerprinting. Microsatellites can also occur in gens, and there are examples where the expansion of such repeats disrupts gene expression and causes disease, for example, CAG, N and polyglutamine repeat disorders, CGG, N and Fragile X syndrome, see Lecture 4. 23 Microsatellites and DNA fingerprinting. The principle behind DNA fingerprinting is simple. DNA samples from two unrelated people differ on average at 1 base per thousand. Since the diploid genome is 6 billion base pairs, this amounts to approximately 6 million differences in the sequence of two people. Identical twins have very similar DNA sequences, though not identical. Why? And the more closely related two people are, the greater the chance that their DNA sequence will be similar. If we can detect these differences, then we have a way of uniquely identifying people from their DNA sequence. We could, of course, sequence everyone's genome, but that's still a bit too expensive at the moment. We can, however, take advantage of polymorphisms associated with simple sequence repeats to assess DNA. Single locus fingerprint gel. If we take specific primers, thicker rows above, that bind to unique sequences flanking a polymorphic SSR, we can use these in a PC reaction. Since the SSR may be a different length on each chromosome, each individual can produce two fragments in a PC reaction. These we can easily resolve on a polyacrylamide gel, which is capable of base pair resolution. If we compare two people, they are unlikely to have the same sized bands. However, the polymorphisms displayed at each polymorphic region are by themselves very common. Typically, each polymorphism will be shared by around 5-20% of individuals. However, if we look at multiple loci, then the unique combination of several polymorphisms in an individual makes FEMethod discriminating enough to be a robust identification tool. The National DNA Database in the UK is the SGM+, second generation multiplex plus, DNA profiling system, which includes 10 different regions and a sex-specific, X or Y, test. The probability of an unrelated match is 1 in 1 billion. SGM plus profile. In this example, each pair of peaks represents Phe genotype at a single locus on each of Phe homologous chromosomes, with the boxed number below the profile showing the number of repeats in each allele. The amylogenin locus gives X and Y specific products of 106 and 112 bits per second respectively, showing that this profile is from a male, D8, D8S 1179, has 13 repeats on one chromosome and 14 on the other, D19 same number of repeats on each chromosome, and so on. 24 segmental duplications, low copy repeats. These are large blocks of genomic sequence sharing a high degree of sequence identity, 95%, that occur in 2 or more copies per haploid genome. Primate genomes are especially rich in these low copy repeats and they make up about 5% of the human genome, with many duplicants being 20 kb. They occur throughout the human genome, with some chromosomes having a particularly high density, for example the Y chromosome. Some are scattered across the genome, others occur tandemly distributed, while some are clustered in 2 zones. One consequence of these low copy repeats is the evolution of new genes and functions. This applies especially to small compact genes. Their duplication has given rise to multigene families. This can be useful to provide an increased level of gene product where it is required. For example, clusters of histone genes are found in many organisms. If we estimate that a single nucleosome occupies about 200 BP of DNA, then we need 30 million copies of each histone molecule each time DNA replicates. That's a lot of histones in a short space of time, hence histone genes are often duplicated to provide more copies. Gene duplication is also very interesting from an evolutionary perspective, since it has been one of the driving forces behind the generation of diversity. Here genes are duplicated and then, freed from the constraints of selection, can acquire mutations and evolve new functions. Duplication of the XQ-linked opsin gene in an ancestor of higher primates, followed by mutation of the duplicated copy, gave rise to the red and green opsin gene cluster, which facilitated our detection of a wider spectrum of light and trichromatic color vision. However, the existence of duplicated, near-identical, DNA sequences also makes such regions of our genome susceptible to structural rearrangement, hence the red-green color blindness trait common in Caucasian males. Other classic examples include the well-studied globin gene family. Both alpha and beta globin genes have been duplicated during evolution and have evolved slightly different functions with specialized genes expressed in the embryo and fetus. Remnants of this evolutionary process can be seen in the degenerate non-functional pseudogenes at both alpha and beta globin loci. As you will learn in the lectures on development, the Hox gene clusters encode a set of related, important genes. Fat control development Hox gene clusters are found in all metazones and are composed of a set of related genes encoding proteins that regulate how sets of genes are turned on in different parts of the developing body. All of the genes in the Hox clusters are clearly related. It is believed that they have been maintained as a group in the genome to facilitate precise control of their expression. 25 gene numbers. As we have touched on, the issue of gene number in a given organism is a thorny one. Ultimately it depends upon the quality of the sequence and its annotation. With unicellular organisms gene numbers are relatively low and the genes are very densely packed in the genome. These organisms tend to divide quickly and hence need to replicate their DNA quickly. E. coli can divide every 20 minutes under good conditions, consequently they have eliminated waste from their genomes. As we reach the metazones things change, genes are more numerous and more spaced out. However, even here there can easily be a 5-fold difference in gene density between related species, cf. rice and maize. In part, genome complexity is due to the fact that individual genes are more complex in metazones and within the metazones mammalian DNA is particularly complex. If we measure gene complexity by the number of exons each gene contains, we find that almost all budding yeast genes have a single exon, 83% of Drosophila genes have at least one intron and 5% of mammalian genes have over 30 introns. Some genes are very complex. For example, the flied scam gene has over 100 exons and the largest intron in the human genome is 480 kilobits, as large as the smallest mycobacterial genomes. All of this together makes the identification and accurate annotation of metazone, and in particular mammalian, genes a difficult task. Until de novo prediction tools improve, we are limited to a set of experimental and comparative tools. We can compare genomic sequence with the sequence of processed RNAs, obtained from DNA libraries or RNA-seq. From the primary sequence of the proteins the genes are predicted to encode and from information derived from simpler organisms. 26 Lecture 3 Summary. You should understand the following concepts. Human mitotic chromosomes can be characterized via their genes-banding patterns, cytogenetics. Functional elements of eukaryotic chromosomes. DNA replication origins are specific sites for initiating replication. Telomeres in immortal cells need to compensate for the incomplete replication of the lagging strand at chromosome end. So, the centromere is the locus at which a large multiprotein complex, the kinetochore, assembles. The kinetochore is the primary chromosomal attachment site for spindle microtubules. The DNA elements required for origin, the Rs, telomere and centromere function in S. cerevisiae can be assembled to generate a yeast artificial chromosome, YAC. Full stop. YACs do not function as chromosomes outside budding yeast. All large arrays, hundreds of kbs, of alpha satellite DNA have been used to generate human artificial chromosomes, HACs, in human cells grown in culture. HACs will not function as chromosomes in budding yeast repetitive DNA. 45% of the human genome is derived from transposable elements, so a small number of line elements are active and create de novo insertions via reverse transcription of RNA. Simple sequence repeats, also known as satellite DNAs, make up 5% of our genome. Some satellite DNAs have functions, for example at centromeres and at telomeres. SSRs are highly polymorphic in populations and are useful genetic markers. SSRs are very useful for DNA fingerprinting. Other types of repeat include segmental duplications. Gene families represent evolutionary duplication of a gene and acquisition of new functions, for example, histones, opsins, globins, Hox gens. Genome complexity. Gene numbers, like DNA content, do not always equate with complexity. Metasome gens tend to be more complex and have multiple exons, so a single gene or genomic locus can generate multiple different functions. So gene complexity makes annotation difficult. Need to make use of all available sources of information, including mRNA sequence, predicted proteins, comparative analysis with other genomes.